Good morning, my peers, and fellow researchers. Today I'm super excited to get to talk to you about what I've been researching on for this past year. Many thanks to the Office of Undergraduate Research for this opportunity and for Horvath Labs. With that, let's get started. So today I get to talk to you about novel constructs for gene editing. So first, I'd like to start off with why is gene editing important? A good place to start is with SNPs. Now, what is a SNP? Well, a SNP is a single nucleotide polymorphism, where a single base changes at a very specific location in a DNA sequence. There is currently 94,651 SNP-related diseases, and that number is currently climbing the more we discover about them. Now, there's very specific SNPs that have to do with changing a GC to a TA transversion that we'll be focusing on for the purpose of this presentation. And there's currently 8,930 of those diseases. And that number will be important for later in the presentation. So, what are some SNP-related diseases? Some you might be familiar with is sickle cell anemia, cystic fibrosis, diabetes mellitus, Huntington's disease, even chronic heart failure. Now we have DNA editing systems that currently exist, but they struggle with A to C edits, which is what we focused on in our lab. But before we get into our DNA editing systems, another point I would like to talk about is the importance of adenine and guanine bases in genetic editing. First, adenine comprises about 31% of all of our DNA bases in the human genome. Guanine comprises approximately 20% and is also the most easily oxidized. Now, why is this important? Well, here we get to talk about my favorite enzyme, MutY. What is MutY? Well, MutY is a DNA glycosylase enzyme, and its sole purpose is to repair DNA by recognizing when a guanine becomes oxidized. It gets mispaired with an adenine, and MutY's job is to remove that adenine so that that DNA sequence can then be repaired rightfully to its original G and C base. MutY is a natural enzyme that already exists inside the human body, and it has this intrinsic ability to already do genetic editing. Now, how does MutY work? There's a lot on this page, but as I said before, the main thing I would want us to focus on is the very fact that MutY recognizes when an oxidized guanine and adenine lesion takes place. It doesn't remove the damaged DNA base. It's not removing the oxidized guanine. It removes the mispaired adenine. So it takes that DNA sequence, it bends the DNA, and it extrudes that adenine into its active site. It removes that adenine, and then it sends that DNA sequence down downstream bare pathways to restore that original C and G. So let's talk about how other editors work now. One is prime editors. Prime editors have what's known as a PEG RNA sequence. This helps to guide the Cas9 to the intended place of the edit. It also contains our intended edit in that sequence. So our PEG RNA with our intended edit is directly fused to Cas9 and reverse transcriptase. Cas9's job is to nick that target strand. Reverse transcriptase then copies the PEG RNA with that edit in, and then downstream endonuclease repair takes place, and voila, you have your edit. Now, how does this compare to base editors? Well, base editors are a lot simpler and smaller in size. They have a guide RNA strand, which is then fused to Cas9 and a deaminase. So the guide RNA takes the Cas9 to where that cut needs to be made, it nicks that strand, and the deaminase focuses on a single nucleotide. It helps to convert that base either from like a C to U or an A to I on a single strand. And then the cell is left up to its DNA repair mechanisms to resolve that final edit, either a C to T or A to G, etc. And there you have an edit. So then this is where I would have talked about our novel construct. But here is where it gets fun. This research is currently under a patenting process, so I actually can't talk about a lot of it, but it's really, really cool. Basically, Vincent Mays, now Dr. Vincent Mays, was a previous PhD student in our lab which created this construct. It was created around the novelty that MutY has the intrinsic ability to already do an adenine-excision naturally. So we took this enzyme found naturally in the human body and made it programmable to make edits. Structure would be really, really cool. It is really cool. So what are some tests that we did? Well, first, we did so many tests. It's been crazy. But the two main ones I'd like to focus on is we did affinity combinations. When creating an editor, we needed to know, can our construct, does it like RNA, DNA? Can it just focus on guanine, or does it need to be oxidized guanine? Our second one was our construct combinations. We made so many different variations of this construct, and we needed to know which one worked the best. The majority of these were tested using RIF assays. So here's some data that we gathered on our affinity combinations. So I put some side notes in case those who don't understand a lot of the kinetics, and that is, in our first row, when we're talking about Km, a lower Km value means that MutY has a higher affinity for it. Here we showed that MutY had a higher affinity for oxidized guanine. Our next column is our Kcat. A higher Kcat value shows a faster turnover rate. Here we showed that it had a faster turnover rate for oxidized substrates. And lastly, a higher Kcat Km value means a higher catalytic efficiency. We showed that it optimized efficiency for oxidized guanine in DNA. So what are our overall trends? Our overall trends is that MutY prefers DNA over RNA and oxidized guanine over guanine. So now we would touch on our construct combinations, all the different ways we've pieced together this construct, and we need to know which one was working the best. Well, you can tell from our value, some had higher net efficiencies than others. Some had higher purity, and that's what we wanted to see. We could then take those constructs and compare them to existing editors. We could test our most efficient editor against existing editors. You can see highlighted in the green in that table that we were having a true efficiency of 2 and 1.4 as our current best performative editors. So let's end with some fun stats. Prime editors currently have 55% efficiency depending on the prime editor. Their advantage is that they can create indels and SNPs, they can do successful editing, they have a greater reach from their PAM site, so the Cas9 for editing has a greater reach. It has a higher specificity and lower off-target hits. That means not making edits where edits shouldn't be. However, its disadvantages. It's generally less efficient. It has higher indels and risk of scaffolding sequence incorporation. That scaffolding sequence has the intended edit in it, but you risk having more of that scaffolding sequence incorporated where you're making that edit. It's a risk. Then we have base editors. Base editors for A to G and C to T, currently 90% efficiency. That's really high. However, their A to C edit that MutY focuses on, they only have around 56% efficiency. However, its advantages. It's more efficient. It's smaller in size. and it has a simpler mechanism. Disadvantages: it has a narrower reach from its PAM site, and it is at a high risk of nearby A and Cs becoming deanimated. That means unintended edits on nearby bases that shouldn't be edited. Then we have our editor. Right now, our stats kinetically are showing a high affinity for OG in DNA. Its potential advantages is high precision. That means it would have low off-target events, MutY very specific. This results in high purity, only intended edit at the edit site. But then we recognize it has potential disadvantages as well. Its complex mechanism could really impact its efficiency, and its larger construct size would make it harder to deliver. Overall, our research underscores the capacity of this construct as an innovative solution for precise gene editing. By integrating a natural MutY into gene editing methodologies, we're improving accuracy, reducing potential risks associated with genomic modifications, and it deepens our insight into gene editing processes, paves the way for significant advancements in personalized medical therapies and genetic interventions. So, its directions: far-reaching medical applications. Like I said, it's an enzyme already found naturally in the human body. This creates a door for the development of more personalized medicine. And we've shown that it could potentially lower the prices for current gene editing treatments. And, as I mentioned before again, there's that 8,900 human diseases that are caused by these C to A transversions that our construct would help to address. Many thanks to Horvath Lab, who became my laboratory and research family over these years, and a big thanks to the Office of Undergraduate Research. For any questions, please email me at u1390144@utah.edu. Thank you.